Welcome to Life and Biology. I'm Dr. Joel Graff. This is a condensed lecture going through some overhead slides I drew for a class on February 18th, I believe. This is the 16th lecture for the class this semester. The title for this is From ECM to ATP. ECM was something I mentioned towards the end of the last video. It was the stuff on the outside of an animal cell. So just to make it real exciting, I called it fun facts. But so let's just run through those. So the first is that there's collagen outside of cells. This is the most abundant animal protein, and it can even be found in fossils. In fact, they were able to demineralize a Tyrannosaurus rex bone, collect the protein that was still there, and send it through what's called a mass spectrometer to determine the amino acid sequence of the protein. And they found that what they were able to find was the collagen. And when they compared the sequence of amino acids in the dinosaur collagen to current modern day animals, it was most closely related to birds. So that's my fun story for collagen, is that we know dinosaurs and birds are closely related, and there's one piece of the puzzle. So number 2, proteoglycans. Basically, I didn't have much to say about this except for I want you to get used to the idea that if you look at the names of different things in the body, different molecules, you can kind of figure out what they might be. For example, proteo should make you think about proteins, and glycans is sugar decorations. So whenever you see glycan, it has to do with carbohydrates. So proteins with some sugar decorations on them are proteoglycans. Now, when a cell goes to secrete a protein, that protein will follow the endomembrane system, which we talked about a couple of lectures ago. And on the way to the surface of the cell, the proteins have to first go through the ER, then they go to the Golgi, then they go outside the cell. Hopefully my little mouse movement helped you picture that. But anyway, the proteins in the ER will start getting some sugar decorations on it, and then as the protein goes from the ER out through transport vesicles out to the Golgi has its own sugar modifying enzymes, and so it'll modify the sugar decorations. That's why I called it decorations, is because you can get different versions of sugars on the surface of the proteins, and then it'll be transported outside the cell. So a lot of times transmembrane proteins are secreted proteins, or in this case the proteoglycans, they're going to have a bunch of sugar on them, and that's from going through the endomembrane system. So that's a tie-in to a couple lectures ago. And then with integrins, these are transmembrane proteins. Now they're folded over when they're inactive, and they usually are folded over below the height of the extracellular matrix, and then they can stand up straight, so to speak, when they become active. And so when they do that, then they attach to other cells, and by attaching to the other cells, you also get these proteins to act as a start of a signaling pathway. And we haven't talked about signaling pathways yet, but anyway, it sends a signal into the cell and starts a communication chain. So that's what these integrins can also do. So those are your fun facts for extracellular matrix. There's more to it, obviously, but that's it for now. Then in class, we watched this video. There's a link to this video. It's called The Inner Life of a Cell. So just go look for The Inner Life of a Cell, and you'll be able to see what we watched. But it covered all sorts of different organelles. It was an animation, and I like showing it each year because we're kind of wrapping up cell structure at this point. You can also see the extracellular matrix in this video very nicely on the very first cells it shows. Okay, moving on. Outside of a plant plasma membrane are the cell walls, and it might be one cell wall or it might be two. So primary cell wall, which is something that all plants have, is going to be fairly thin for the most part. But then some plants will have secondary or thicker cell wall. So in the drawing, the black line here is the inner or the plasma membrane. The red line that's on the very outside is the primary cell wall. And if you have a plant cell with the secondary cell wall, it'll be situated between the primary cell wall and the plasma membrane. It's an optional thing, and where you're going to find these thick secondary cell walls are in woody plants. And as you can imagine, if you are a plant like a tree that's going to grow tall into the air, you would want to have a lot of structure, strong structure, and so this secondary cell wall is part of the reasons that things with wood in them can grow so tall. Okay, well that's it for different cell parts and all that. We got all the way to the outside of the cells. So we're going to move on now. We're going to move into metabolism. Metabolism can either be breaking or building up of molecules, which would be anabolic, and you need to put energy into the system, and one energy source would be from a chemical called ATP, a molecule called ATP, or you might break down molecules. Those are called catabolic pathways. And when I say molecules, I might also say metabolites. So the different molecules that you find within a metabolic pathway could be called metabolites. So generation of an ATP is going to be one of the goals of photosynthesis, and we're also going to try to generate ATP when we are breaking down the glucose that was made during photosynthesis. And so what is ATP? Well, it's adenosine triphosphate. So adenosine, I'm not going to draw the structure out here. We never bothered when we were learning about the nucleic acids what adenosine look like. You can Google it if you're curious. But off of adenosine is going to be a string of 3 phosphates. These 3 phosphates are, like most phosphates, are going to have negative charge to them, and there's going to be built up chemical energy here. And if we're going to use up an ATP, get some of the chemical energy and convert it to hopefully kinetic or some other useful energy, or maybe just some heat, we will break off one of the phosphates, and then you'll end up with adenosine diphosphate, di for 2. And because you broke off the tip, the other molecule you end up with is just a free-floating phosphate group. That's called inorganic phosphate. And also you get some energy released. And then I drew this over here. Let me just delete that. I was just drawing another shorthand version of phosphate, but I could see where that could be confusing on this slide. So ignore that. But to break the bond here, we're breaking a covalent bond. And like so many of the reactions we're going to see, if you're going to break a covalent bond of a molecule down to smaller molecules, it's going to often be a water molecule that is going to be added into the situation so that you can satisfy the phosphate that's left without oxygen after that break. Okay. And so then, of course, energy is released, and it just happens to be the right amount of energy to do stuff in a cell. So because ATP is going to be the general size of an energy packet, a lot of things in the cell are going to take an ATP to get the job done, or for each step of a process, you might need an extra ATP thrown in. So anyway, that's what these metabolic pathways, we're going to keep coming back to ATP. So here's just a nice little drawing of how you break the 3rd phosphate off and you get some energy out of it. Occasionally you break 2 phosphates off instead of just 1. In that case it would be called a pyrophosphate, but you don't really need to know that. Okay, so then what well, how much energy is in ATP? So let's say we have one molecule of ATP, and we're going to set that equal to 1 ATP worth of energy. And we're going to want to know how much or how many ATP worth of energy we're going to have in different molecules that we're going to see. So as we go through this list, we first have FADH2, and that molecule carries a couple electrons that have about 2 ATP worth of energy. NADH, those have about 3 ATP worth of energy. NADPH have about 3. Really, the only difference between NADH and NADPH is a phosphate group. And don't worry about what the NAD or the FAD looks like. Just know that we're going to have some of those coming into play when we go through some of these metabolic pathways. The pyruvate that you get once you break a glucose in half is worth about 10 ATP. Okay, and then glucose, that's the molecule, that's the energy molecule you build up in photosynthesis and break down in cell respiration, or at least they're the classic thing that gets built up or broken down. Those have about 100 ATP. So without oxygen around, you only get 2 ATP per glucose. So of the 100 possible ATP chemical energy that's built up in a glucose, you only get 2 ATP. And so the other way that we can break down glucose is in the presence of oxygen. And in that case, if we have oxygen around, you're going to be able to capture about 32, give or take a few ATP per glucose molecule. And this tops out at 32, or the other choice without oxygen is 2, and this all seems very wasteful, but in class I pointed out that even car engines, when you're burning some gasoline, you usually only get about 30% of the energy from the chemical energy from the gasoline is converted into kinetic energy. Other energy from the gasoline is going to be released as sound, and also, more importantly, a lot of the energy is going to be released as heat. So anyway, now we're all the stages set to go into photosynthesis in the next lecture. All right, this has been Life in Biology. I'm Dr. Joel Graff, and you can like and subscribe or not.